everyone. Topic for today is gene therapy. Gene therapy is the intracellular delivery of genes to generate a therapeutic effect by correcting an existing abnormality. The Human Genome Project provides information that can be used to help replace genes that are defective or missing in people with genetic disease. Gene therapy was once considered a fantasy. In 1990, a 4-year-old girl was the first patient to receive gene therapy for severe combined immunodeficiency. She became a healthy adult with an immune system that was able to fight off most of the infections. What are the outcomes of gene therapy? It replaces a mutated gene with a healthy one. It deactivates a gene that is not functioning properly, introduces a new gene into the body to help fight off the disease, enhance the effect of a normally functioning gene, activate the gene that was shut down during the fetal life. Based on the purpose, gene therapy can be classified into gene replacement therapy, gene deactivation therapy, transgenesis, gene enhancement therapy, and gene activation therapy. Where do we introduce these genes? Somatic cells or germ cells are the cells to accept these introduced genes. Based on the type of cells involved, gene therapy can be somatic cell therapy or germ cell therapy. Germ cell gene therapy means introduction of functional gene to the germ cells. The change produced will be inheritable, and this is not being investigated in humans as this is considered as unethical. Somatic cell gene therapy means insertion of fully functional and expressible genes to somatic cell to correct a genetic disease. Here the change produced is not passed into the next generation. How do we introduce these genes? There are 3 ways: ex vivo strategy, in vivo strategy, and in situ strategy. In ex vivo strategy, the patient cells are cultured in the laboratory. The new genes are infused into the cells and these modified cells are administered back to the patient. In vivo strategy, the vector is administered directly to the cell. And in situ strategy, the carrier of the gene is injected to the patient either intravenously or directly to the tissues. So in ex vivo strategy, the copies of the therapeutic gene is inserted into the viral DNA. Then target cells, it is removed from the patient and it is grown in a culture. Then these cultured cells, they are infected with this genetically altered virus. Now the target cells are genetically altered with therapeutic gene. Then these cells are reintroduced into the body. That is the ex vivo strategy. And in vivo strategy, the therapeutic genes, they are inserted into the viral DNA. Then this genetically altered DNA is inserted into the patient body by cell-specific direct tissue injection. Then this DNA is incorporated into the cells and they will produce proteins which is encoded by the inserted genes. That is the in vivo gene therapy. Summary of the procedure is: isolate the healthy gene along with its regulatory sequence, incorporate this gene onto a vector and deliver the vector to the target cells. Vectors are the carrier particles or molecules which is used to deliver the therapeutic genes to the target cell. Different vectors used include viral vectors and non-viral vectors. And why vectors are needed? Since the genetic material has to be transferred across the cell membrane and preferably into the cell nucleus. So the process of virus-mediated gene transfer is called as transfection. And how do we select these viral vectors? It depends on which cell they can recognize and infect, how well they transfer the genes to the cell, whether they alter the cell's DNA permanently or temporarily. The commonly used viruses include retrovirus, adenovirus, adeno-associated virus, herpes simplex virus. These are the commonly used viruses. Retrovirus, they are RNA virus with reverse transcriptase activity. So the commonly used one is mononemurin leukemia virus. The gag, pol, envelope genes, they are deleted from this viral genome so that it cannot replicate inside the human body. After deleting these genes, desired human gene is incorporated into the virus, which is then cultured in a media containing packaging cells. These packaging cells, they will provide necessary protein to pack the virus. Then this virus, it is introduced into the patient, enter the target cell and carry out reverse transcription. The complementary DNA so formed is incorporated into the host genome and it gets expressed. As we said, the 3 genes of viral genome are removed. Then the desired segment of DNA is inserted. Now a mutant virus is formed. Same thing, delete gag, pol, envelope genes. In that place, we add the desired human gene. Then the viral RNA has to get a protein code to enable them to enter into the human host cell. So this defective virus is put into packaging cells which will provide the viral code. Then this virus particle with protein code, they are infective but they cannot replicate or produce disease. Then they will enter the host cell by receptor mediated entry. In the cytoplasm of the human cells, the reverse transcriptase carried by the vector will convert RNA to the proviral DNA, which is then integrated into the target cell DNA and the normal human gene can express. The advantages of retrovirus are the virus is replication deficient, so it is safe and is suitable for the treatment of a variety of disease. Disadvantages, random insertion can disrupt normal genes and retrovirus use rapidly dividing cells as the target. So the non-dividing cells cannot be used. Adenovirus, they are DNA virus. The virus carrying the human gene reaches the nucleus of the target cell and they remain epichromosomal. They do not integrate into the chromosome. Advantage of using adenovirus is it can infect a wide variety of cells. And the disadvantage is expression is only transient. So the vector, it binds to the cell membrane. It is packed in a vesicle and the vesicle breaks down releasing the vector. And the vector, it will inject the new gene into the nucleus and the cell makes protein using this new gene. This is gene therapy using a adenovirus vector. So the non-viral vectors include, that is the vectors other than the virus. It include liposome, plasmid and the physical methods. Gene gun method is a physical method. Here the tungsten particles are coated with the plasmid DNA and it is accelerated by helium pressure discharge. So this enables the particles to penetrate the target tissue and gets incorporated into the genome. Advantages, it is quick and can be used in all tissues. Disadvantages, it causes cellular damage and transient gene expression. Next one is plasmid liposome complex. So what are liposomes? They are artificial lipid bilayers. So these liposomes, they are incorporated with plasmids carrying the normal human DNA. So this plasmid liposome complex, it can enter into the target cell by fusing with the plasma membrane. Advantages, it can carry human gene of large size, they do not replicate and they will evoke only very weak immune response. Disadvantages, most of the complex, they are destroyed inside the host cell. So plasmids carrying the normal human gene, it is inserted into the liposome. The plasmid liposome complex, it will enter the host cell by fusion of membranes. It will not enter into the nucleus but it remains epichromosomal and there will be expression of the normal gene. These are some of the examples for gene therapy trials conducted. Severe combined immunodeficiency, ADA gene in chromosome 20 was transferred using retrovirus. For cystic fibrosis, CFTR gene on chromosome 7 to bronchial epithelium by adenovirus. Hemophilia, A and B genes for factor 8 and 9 into fibroblast by retrovirus. The accomplishments of gene therapy include severe combined immunodeficiency, which is caused by the deficiency of adenosine deaminase enzyme, The first trial of gene therapy was done on this disease, and the follow-up studies showed the presence of normal immune function in recipients. Then activation of HbF gene in patients of thalassemia and sickle cell disease. These are some of the accomplishments of gene therapy. The drawbacks are the short-lived nature of gene therapy, immune response to foreign object introduced into the human tissue, chance of inducing tumor by insertional mutagenesis, and death during the course of gene therapy for ornithine transcarbamylase deficiency was reported. Transgenesis. The human genes, they are micro-injected to farm animal and transgenic construct have been produced. So the transgenic pigs, they produce human blood which is suitable for transfusion. And transgenic mice, they produce human hemoglobin with the same oxygen carrying capacity. Anti-gene therapy is the treatment of disease by blocking the transcription process using single-stranded oligonucleotide sequence that hybridize with specific gene. Antisense therapy, it will inhibit the translation by single-stranded nucleotide. That is, mRNA contains a message to be translated into protein. If an oligonucleotide chain having complementary sequence to mRNA is made, it is antisense. When an antisense oligonucleotide is added, what happens? It will trap the normal mRNA and protein synthesis is stopped. Antisense nucleotides are delivered into cells by liposome encapsulation. Clinical trials on HIV and cancers are being conducted using antisense molecule. Now, gene library. Thousands of genes arranged in specific order in the entire length of DNA. That is, DNA of an organism is cut into small pieces of about 100 kilobase pair by different restriction endonuclease. These cut pieces are then inserted into the vectors and amplified in bacterial cells as recombinant DNA. A collection of these recombinant clone is called a gene library. That is, the human DNA using restriction endonuclease is cut into DNA fragments. These DNA fragments are inserted into plasmid using ligase. Then these plasmids are introduced into the bacteria. That is gene library. Thank you.